Okay, so we're going to start talking about our fifth session in the lab, in which we get to put several labs kind of going side by side. That means that we're looking at a little bit of a discussion on our pre-lab training. I'll mention that to begin with. Then we'll set up our lab 19, which is starting a little out of order for our labs, but for the sake of efficiency, we're starting now. And then we'll talk about the instructions for lab 6, and then we'll kind of conclude everything with your familiarity with microscopy and actually using the images that we're going to generate during the lab here a little bit later. So that's the plan. Now, that being said, let's kind of start off with our pre-labs first. Now, several of the labs, just like similar to what you've done in previous labs in biology and in chemistry, the requirement is for you to come prepared prior for you to doing the labs. This helps to reduce several things from cheating to reducing people taking advantage of others and that kind of thing, so that everybody shows up prepared and nobody's kind of bugging anybody else to know what to do. That's usually part of the principle behind that. But for us in microbiology, it also prevents people from getting hurt by actually knowing what you're actually going to be working with during those labs and knowing the safety measures and so on and so forth. It brings overall a more efficient lab as well as a safer lab altogether. Now, the instructions on how to run a pre-lab are actually run from on Canvas. They're posted on module 1.2. And there's this document, this PDF file that actually has 3 different versions of how to write a pre-lab. So let me share that with you really quick. Let me actually select the screen. And if you go into your course modules, you'll notice that in module 1.2, you have this little document that says pre-labs, right? So if you click on that bad boy, what you'll see is a 6-page document in which the first 3 pages, it's in detail, multiple details put into how to write a pre-lab, the importance behind it, how to write it, what's its content, and several different examples into it. So the first 3 pages is a very long, drawn-out discussion on how to write a pre-lab. Now, if you already are a little bit of an expert, you're familiar with how a pre-lab works and why, there is a second version of it called the shorthand ones, about 4 pages in, actually, that says the shorthand guidelines. And so this is for people that already are familiar with it, just need a little bit of a refresher just to know what they're going to be working with, including format. Now, if you're already fairly familiar with the system and you're good to go and you just want to know exactly what needs to be contained, there's a really, really, really short version of it right at the end of that one, in which is just 5 or 6 lines in there, and that's it. So that means just the instructions that you need to know. Now, that apart, I do provide extra examples right after it as well. And then I also provide you with a grading rubric. So when I'm grading these guys, or the graders are taking a look at it, there's a series of comments, the most common ones that pop up on, are you missing something? Did you add something that didn't need to be included? And so on and so forth. So all of it is included in there as a way for you to have a reference as well. And then the last thing you can have in here is how to take care of the scope. So once we get to the scope portion, we have some instructions on how to properly maintain them. So they're part of the same document in there. So that's where we're going with that, right? So let me go back to our presentation here. Our very first lab that is going to require pre-labs, as you can see from our syllabus and our schedule, are labs 12 and 13, which are going to be the motility labs. And so that will occur the following week. So we still have a little time to work on them. Now, because the first set of labs that require stains, lab 14 and 16, are coming up in the following session, if you'd like to have a little bit of practice, you can always set some of these up and send them to me via email, not on Canvas, and I can actually grade them for you just if you want to kind of get familiar with the process. Now, the format is, as usual, it's simple, it's typed, it's short, it's efficient, but for those of you who want good practice in a good lab notebook, you should include them in your lab notebook, handwritten as well. So this is the idea behind repeating this a couple of times for better studying. As you're going to find out a little bit later, the pre-labs are going to be a key component in your second and third lab practicals, as they're going to be part of the material that you need to learn for them. So this is why practicing the pre-labs now pretty much sets you up in a good start at this point in time. Now, when you submit them, they will pop up as portals, usually on where the same place you'll find your lab reports. You'll see a portal that says submission for pre-labs, And the idea here is, again, without the pre-lab, you're not going to be able to complete the lab. That's part of the rules that were written in the contract. So you'll have a portal, in which you'll submit a typed in page or pages containing the pre-labs that are pertinent to that particular lab. Submit them, we will grade them, and that will give you permission to access the lab portal for you to submit the reports. So that's the overall breakdown of it. So you should have already read this material, should be familiar with this. Again, most of you have taken chemistry, if not all of you, and should have this background already. The only difference for us, they're a little bit more abbreviated, and they're mostly focused on just key steps and safety. Those are the overall premises of our pre-labs. That aside, let's make sure we can take a little bit of time to answer any questions that we have for this stuff. Okay, so moving on from our pre-labs, the next piece we need to deal with is the actual lab itself. So here we're actually setting up a lab that we're not quite going to focus on until the 4th lab report. Now, the reason why we're doing this at this point in time is to be a little bit more efficient, as we're trying to save on organisms and having to have you guys come into the labs again for no particular reason. We're giving you the opportunity to grow these things now. And that's what we get to do with Lab 19. So Lab 19, the main premise behind this is selective and differential media. And what you're doing really here is learning the properties of the organisms you're studying. So part of what I'm trying to promise here is you now get to observe and really find out what you're actually testing. And so what we're going to take here is, rather than us providing you with an original sample, something that we grew in the lab, you're actually using your own stuff. And what you're going to be doing is 2 things. One of them is going to be observing it for pigmentation, and we'll start with Lab 6 there. And then we're also going to be looking at its ability to grow in certain types of very specific media. This is the S&D portion of Lab 19. So that's the overall premise. So let's go with some detailed instructions. The very first portion is Lab 19. So we're going to start with Part A. And so here what we're going to do is we're going to set up 2 new plates. This is going to be our MSA and EMB plates, our mannitol salt agar, and our eosin methylene blue plates. These are the smaller plates, I'll show you in a moment, that all you're going to do is do a quick inoculation. You're going to still use your loops. You're going to do little fish tails in the center of them, and we'll be done with that. Now, your source should be your broth. That same broth that we've been maintaining for the last couple of days in the previous session, that's the guy you're going to want to use. Now, in the event that that guy didn't work, that did not grow enough, remember I told you to have a backup plate just in case, one of the streaking plates that you did, so you can use that as well. So if the broth doesn't work, resort to the previous plate. Now, what's going to happen is we're going to actually set this up prior to the next session. So that means that you're going to be able to complete this one before we meet again. So what's going to happen is you're going to inoculate, let it sit for about 48 hours. Sometimes it may grow a little bit earlier, or sometimes it may take a little bit longer, but it really shouldn't phase more between 1 to 3 days maximum. What you're going to do, inoculate it, allow it to grow, and then as usual, collect, observe, take pictures, and annotate what you've observed, just like we've done everything else in your lab manual and your lab notebook. The only difference now is that this is going to take place prior to us meeting for the next session. Now, this will be part of your lab report 4 because it's Lab 19 and that's part of that series. But because we needed something growing to test, we're starting it now. This is where the time differential just kind of is a little bit off. So let me show you what we have really quick. Let's kind of switch cameras around. We're going to take our lab kits that we had before. I'm getting my gloves on so we can play with these guys. And let's show you what else we have left. Let's go with this guy. There we go. So let's stop this for a moment. There we go. So for those of you who remember this, let me see if I can get a better close-up of this guy. Here we go. This one should work a little bit better. There we go. So in your kits, you should still have your little Ziploc baggie that was part of the larger one. And you should also have remaining one plate that was our spare plate. So this guy is just kind of holding there just in case things may take the wrong turn. So out of our little baggie, what you should have remaining in there should be 2 small plates and then a very similar looking plate to the other ones, but this is a Mueller-Hinton plate. This is designed for antibiotics, so don't mix these guys up. Now we're going to take the 2 small plates out. 2 little mini plates. And then we're going to leave the other guy in its Ziploc. Again, this one's, even though they look very much the same, this is actually a different plate, so don't mix those up. And then the next thing you need is that you should have about 2 of these guys left. So we're going to take 1 more of our little inocula. Okay, that's all we need. So we're going to close this bad boy off. And then we're going to need as our source our little tube, our little broth that you've been maintaining. Okay, so this is the guy. So you may need to resuspend it by kind of shaking it a little bit. You can, again, kind of froth it in your hands to kind of resuspend it. So it should still look nice and turbid, especially if there's stuff sitting on the bottom of the actual tube itself. So what are we doing? This guy off the side. Take 2 plates. Here they are. Right? And then using our standard inoculation loop, right? The guy you got playing with. We're going to use the large loop of the side. And what we're going to do is pretty much kind of do a pinky hold as we did before. Dip it inside this guy really quick. Close it again using the pinky technique. And then we're going to do 2 quick fishtails. All right. Open this bad boy up. Quick fishtail inside of it. Done. Move on to the next plate and do it again. Again, reminding you that you want to have your hand nice and stable, not suspended, so you don't actually stab the auger by accident. Once you're done with the loop, you can actually discard this guy. Now, just in case, I actually did do a little bit of a drawing behind one of these, so you can see what pattern I'm doing. Right? It's just a fishtail. We're not doing any streaks or anything like that. Just a quick kind of painting in the inside so that things can grow. Once you have both plates inoculated, stack them, flip them, let them grow. That's it. This is lab 19. So you're transferring from your original broth, your TSB. Now, again, in the event that this guy did not grow for you or something happened, you should be able to use your previous plate. Same premise, use your loop, fishtail it around this guy, and you should be good to go. Okay. So now that we've inoculated both of these plates, stack them and we'll let them sit there just like we did before. You want them to grow for about 48 hours. Now you may notice some initial growth within 24, and depending on the growth conditions, it may take up to about 3 days maximum. Now what's unique about these plates is that these plates are meant to work collaboratively. One of these plates should be growing while the other one shouldn't, meaning that you should observe growth in one of them, but not the other. And it really depends on which sample do you have. By reading lab 19 ahead of time, you'll be able to kind of determine how that works. And so that's those properties that we're working with, trying to identify what we have from our own samples, what's growing in our broth or possibly our plate, right? So let it grow for a couple of days. And then once they've grown, again, the same idea. Collect it, take images, compare the before and after, but reminding you again that the data that is here is for a different lab report. We're just getting these out of the way so we don't have to come back into the lab and pick up more samples. That's the plan behind these guys. So now let's move back to the presentation itself. There's that guy. And so again, to reiterate here, is the whole premise is we're going to set up lab 19 by using 2 of the mini plates, the MSA and the AMB, and taking it from our original source, whatever your cooties were originally that you picked up, transferring them onto both plates. That's at least a plan for that. That being said, then let it sit there for a couple of days. find what's growing in there, and then go from that point in time. That's it. Now, just to make sure, let me kind of focus again on those plates just one more time. Let's see if we can get this guy going. Wrong one. There we go. On these plates, it's important to know which one is which. And so I have them in my order on my screen over here, but the lighter one of the two is your MSA plate. The darker one of the two is your EMB plate. So the left one on my screen currently is MSA. The right one on my screen is EMB. And if you actually look at them a little bit more up close, you should be able to kind of tell the color apart. The MSA is usually much lighter. The EMB is a much darker plate. So let me see if I can remove this from the background. You should be able to kind of tell them apart. One plate lighter, the other plate is a little bit darker in terms of colors. This is our MSA. This is our EMB. Inoculate both of them, flip, and you should be good to go. Wait 48 hours for them to grow. Take images, describe what you observed, but we'll input that in a later lab. So that is lab 19 for us. Now, as we're going to want to be able to continue with the following session, you're still going to want to maintain your culture. Ideally, these two plates will still have organism in it, at least one of them. However, since we may need to use additional sample, you're still going to want to maintain this guy, okay? Or your alternative plate that you use instead of the broth. So you need to have an organism for the next session. So make sure that you do not discard these at least until we'll complete the next session's lab. So keep that in mind. So now that we have figured out lab 19, the next thing we want to focus on is lab 6, which again is about properties about growth. And in this particular case, color, pigment. So we're actually going to be able to complete both parts at the same time when we run this lab. And so for pigmentation, we already have set up the video for this related on YouTube. So you go for that site. And then we also have the notes and handouts related for this on module 1.2, also on Canvas. So let me show you both in a moment. So let me share the screen really quick. And so if we go onto the DXM page, you'll notice that we already have a lab that says remote medical micro under pigmentation. And this is pretty much just the video recording the exact same samples that you need to input into your lab manual and lab notebook, right? And so we describe what it is, and then we kind of cover each individual one of them. And then as we proceed further into the video, you'll kind of have much better close-ups of them. So you can actually observe the color live. And then, as we explained a little bit earlier, if you go onto the same location where we found our pre-labs, we should be able to go to the next one that says Laboratory 6 images. And so in there, same premise, now you're going to have high definition pictures of that video. So it states Laboratory 6, and there's an overall picture of all the pigments that we're interested in looking at. And then as we proceed further, we'll start zooming in a little bit more and more so you can see the texture and truly compare the colors that they're producing. So that's the content that you want to obtain from that particular handout. Okay, so let me go back. Here we go. So what are you doing here? For lab 6, your job is to identify, to observe the properties of pigmentation, the colors basically, but also understand why. Why is it important to understand pigmentation? For the reasons of it helps us classify things by telling them apart more or less. And key factors in determining pigmentation are based on things like temperature in the environment, salinity presence, something we were discussing in lecture, as well as sometimes even the food that they consume, part of the metabolites that they need to obtain. So availability of light also plays a role behind these things. So those colors that you observe usually are a result of something that's taking place in its environment. So that being said, you can watch the video, you can use the handout and pretty much answer the questions that are in lab number 6. So fill it up in the lab manual, fill out the table as well in your lab notebook, and then don't forget to include the drawings of what's present in the handout, or if you want to do it off the video, that's fine as well. That should complete Lab 6 fairly well. So now we have the final step in today's lab is going to deal with microscopy. So let's kind of go through the details behind this guy. Now, what we want to do is know several things, not only to prepare you for the exam, so we want to review how to use it, how to transport it, how to keep it clean, so part of the maintenance, but also some of its properties. So we're kind of going to go one by one with all of these to demonstrate part focality, as well as final steps in how to use oil to observe things up close. So we're going to have a demonstration for that. That being said, we will provide it with the images that we would generate here. We'll post them on Canvas, so then you can draw them yourself and introduce them into your lab manual and your lab notebook. Okay. Now, the last piece that will be part of next week is you'll be able to use those images to practice measuring things at a smaller scale. And that information is already available on your lab supplement. So if you look that up, it'll show you a little grid, more or less how to calculate under each objective and so on and so forth. So that's the plan for that. Okay. So with that being said, let's kind of move on strictly to just the actual scope itself. So one of the first things we get to deal with is obviously these particular machines are fairly expensive. So obviously we can't send everybody with one as much as we would love to. So you guys had a smaller version of them, a less resolution with them, which are your full scopes. But the premise is still there, being able to generate your own slides and be able to observe them under microscopy to see them a little bit better. But for now, let's make sure we can become familiar with the actual scope itself, the things that are going to be on your exam, and how to use it. So let me change the camera really quick. And now here, what you're going to see is kind of our main side that has the light, the actual switch on and everything else. And I'm going to push this a little bit further back from me so you guys can see a little bit better, right? And one of the things we're going to be discussing is how to use the objectives, how to measure with them. As you can see, the cables are sticking out simply because that'll allow us to use the camera, right? But the main thing you want to learn how to use is the stage. The stage, which is typically driven by the coarse objective of coarse knob over here, should be able to lower it and raise it as needed. And it should only be used when you're under the red or scanning objective, the 4X objective itself. That's one of the main rules. The reason behind it is actually quite simple. Once you actually raise the stage all the way up, one of the things you're going to notice is that no matter how far it goes up, it'll always leave a little gap in between the objective and the actual stage. So this is the only state in which it will prevent the actual objective crashing with the actual stage itself. If you move to any of the other objectives, you'll actually risk breaking the slide and including scratching the objectives themselves. So this is the safe zone, if you will. We always start with the 4X, we always end with the 4X. So we always start with the scanning objective, we always end with the scanning objective. So that's part of one of the main rules on how to use these guys. Now, that aside, one of the other things we want to do is make sure we actually can center our slides and observe under this guy, right? So one of the trainings is making sure you know how to position your slide in here. And so I'm actually going to move this guy around just a little bit to kind of show you a little bit face to face. And to show you how to use the stage clip, because it's spring loaded. And one of the most common mistakes that students make is releasing this guy. And when it releases, it actually cracks the slides. So it's important that you kind of treat this guy gently. So we're going to use our very first slide for today, which is going to be part of just the training so we can have a little bit of fun. And you're going to see me load this guy. You'll notice that I'll open the stage clip. I place my slide in there. And again, I don't release it. I just kind of carefully allow it to adjust onto it. So without doing any damage to it, right? Let me turn this guy on. You'll see that the light turns on. That way you can see it. And we're going to center this guy using our stage adjusters. It's actually quite centered already, probably because I was messing with it earlier, right? Now, the thing is, one of the most common things that happens is, let me switch my camera really quick, is that most people, when they're trying to learn how to use the scope, is that they're looking for their sample by literally just kind of digging in their eyes into the oculars without actually looking at the stage. And what you're now doing is effectively endangering your slide and your scope. So one of the first things you need to learn is how the scope really works. The scope, in fact, is just a couple of lenses put together that you can manually adjust. And very similar to what you can do with your phone, you can actually kind of cherry pick what you want to observe. And more than anything else, you can choose the depth of field. In other words, you can choose the distance between what you're trying to observe and everything else around it. And so just like if you were to take a picture on your phone and you want to kind of blur out the background or the front and only kind of target one thing, that's exactly what the microscope does by selecting a specific depth of field. But in order to find that spot, one of the worst things you can do is, again, put your eyes down and then allow the stage to crash or break the slide and so on and so forth. So how do you actually do that? Well, it's math pretty much. Each lens is measured accurately, is designed accurately to find that depth of field. So without actually having to look into the scope, you can actually find the slide that you need to observe without risking looking through the oculars and crashing something down. So that's what I want to show you with this. So I'm going to switch the camera really quick back again to the other perspective. So here we are again, and I'm going to lower the stage all the way down. And like I said, most people will be right here in the oculars trying to find out where it is. And while they're raising it, we're not paying attention and hoping that they find an image. And then they just end up scanning for a long chunk of time. So believe it or not, all scopes, again, are calibrated, which means that there's actually a trick for this. I'm going to show it to you how it works really quick. The way it's done is actually quite simple. What you want to do is with your actual stage all the way down, and again, making sure you have it on the 4x objective, on the scanning right objective, you're going to raise the stage all the way up because it doesn't crash. And all you're going to do is do a quarter of a turn down. That's it. Just a little bit of a turn on the coarse knob. That's it. And you should have your image available to you. Again, it's already designed to work that way. So let's take a second so I can show you. I'm going to show you a new camera. See if I can find it. This guy's not listening to me for a second. Hang in there with me. I need to disable my background. There we go. And let's make sure. All right. So let's enable this camera. There he goes. Here's the actual view under the scope. So let me center it a little bit. May need to center it. And please note, especially because of this scope that is completely facing away from me, there was no way I was looking under the actual oculars. So this is just by training. So let me kind of switch back again. Right? And so notice that the actual scope is in front of me. I can actually look at it, and you should be able to switch back. Whoops. And see the image again. Not that one, this one. There's your slide. And so with a minimal adjustment, you can get this guy to kind of focus a little bit. And now we have our classic slide of the 3 strings. And part of your exercise is to determine which of the 3 strings is actually on top, which one is in the middle, and which one's at the bottom. So we'll do a little bit of practice in a moment, right? So again, let me kind of show you how this actually works. So let me start with my background over here. As you can see, the strings, and then myself. So let's take this down to the other camera. Now it's having some fun with me. There we go. Let's disable that. For some reason, it doesn't really like the backgrounds. There we go. I'll bring it all the way down. All the way down. All the way up. Small little turn. That's all it takes. And then let me switch the camera over here. And there's your actual focus. Again, notice that I didn't have to observe anything. I just had to follow the rule. The stage raised all the way up, small little turn backwards, you should be able to find your organism. This is done because of how the scopes are calibrated. And so this becomes incredibly helpful when you're trying to look for things that are significantly smaller than this, like bacteria. Most of the time, most students spend a long amount of time looking for things and they're not sure what to find. They're not sure even what they're looking for in the first place. So as long as you follow this formula all the way up, a little bit of a turn down, quarter of a turn down, you will always have your sample as near as focused as you can. Now, the next thing we want to show in here is how to transition from one objective to the next. And for this, we have this little black rubberized piece called a revolving nose piece that allows us to actually move the objectives around without actually touching the objectives. One of the most common things that happens if students try to grab onto the objectives and accidentally break them off or miscalibrate them, which is usually the smallest of the infractions here. So to prevent any of that, use the revolving nose piece, this little black rubberized piece. And so with that, we can literally just kind of turn. We're going to go from our 4x to our 10x objective in this case. So that's our low dry, the one that with the little yellow band, right? That magnifies a grand total of 100 times when combined with the oculars. And let's show you that sky. Now you can see, also still in focus. Now maybe a little bit off, so I'm going to move it a little bit more. There it goes, so I can center it. And so one of the rules that you get to learn about microscopes is once your first slide is in focus, the rest of the objectives should be nearly in focus as well. This is something we call parfocality. And so as you can see, we can already see our two of our strings. And if we kind of play around with it a little bit more, you'll be able to see the other one as well. And so again, part of the exercise, we'll be able to discern these guys back and forth. So we're going to do this again. You can see the yellow string back there too, right? So we're going to do this one more time. Let me switch off. Right, same idea. Using the revolving nose piece, we're going to go to our high dry, the one with the little blue band. This is a grand total of 40 magnification combined with the oculars 10 more times, makes it 400. And let's take a look at this guy. Again, it should be already in focus or near in focus. And let me see if I can focus it a little bit more. Here's our strings. You can see the red one and now the yellow one neatly zoomed in. Let me see if I can go to the blue one really fast. There it is. Ready to go. So that's part of that practice. Now, we should be able to do this again, but we're going to move on to our next target. So let me kind of switch around. Let me go back to this guy. All right. And so what we do, by practice, we always go in reverse. Back to our yellow. Back to our red. And then, only then, we can actually lower our stage so we don't risk breaking anything. So let's take out this slide. And we're going to use the next one in our little repertoire. And enter, probably the most infamous one of them all, is the prepared letter E's. And this one I need to adjust a little bit so I can find it. Should have it centered over here. There it goes. I think I got it in there. So same premise. We learn, raise the stage all the way up till it hits, and then just a slight turn down. Now check it with the actual camera. And there's your letter E. So we already have, let me see if we can center it a little bit better. Letter E, there it goes. Let me adjust it, make it a little bit sharper. And you can actually see the E. Notice that it's a little bit more inverted. For those of you who remember your good old physics days, when you pass light through a lens, it kind of inverts images sometimes. This is what we're seeing here. Now, I'm not gonna change the camera. I'm simply gonna move on to the next objective. Zoom in a little bit more. And again, still in focus with just little to no adjustment over here. There it is. And then we're gonna go to the high dry, our blue one, our 40x magnifying total 400. And now what you're seeing is pretty much the fibers and the ink on the letter E and the little piece of paper. So this is part of what you're going to be drawing into your lab manual and lab notebook. And so then again, we're going reverse. This is from the high dry to the low dry to the scanning, all within focus, hence the term parfocality. So now let me switch. Same thing. So now what we're going to do a little bit is trying to find our next organism. So here we have a prepared slide that contains bacteria, a little bacterial smear. So the same premise, let me switch off my camera over here. The idea is to also set up our organism, put it under the scope, look for it, same rules that apply before. So let me take out the letter E really quick while I'm adjusting this. And we're just going to place our organism in there gently. So let's see if we can find stuff. And I've already have it set. So let me kind of scan around. You can actually see a couple of little dots in there. You can see all those tiny little dots on the camera. That's our bacterial smear, right? Clearly this is under 40x total magnification, but even in there, I'm going to kind of fine tune a little bit. You can see that there's stuff in there, things that are, in this case, millions of times smaller. So we can move to our next objective using a revolving nose piece. And again, part focality tells us that it should be already near in focus. So I'm going to just do a minor adjustment over here, right? And you can now start seeing some of these little strings in there. So we're going to go one more time. And now you can actually see our organisms side by side over here, right? Now, this is part of the training that everybody uses to pretty much identify most organisms, at least their shape, and if you've done a staining, kind of the color that goes behind it. Cool. But this is not where it ends. So let me switch back to the facing camera because we're going to do one more step. And that is the step on how to use the oil immersion. What happens here is that the next objective, the 100 magnification objective or the total magnification of 1000, is actually an oil-based lens, meaning that it requires oil to be used for you to be able to see it accurately. Part two also is the fact that this objective, if you turn it, it will actually crash with the slide. It will actually break it. So in order to prevent that, we add oil so it can kind of act as a lubricant and allow the lens to kind of glide over it without scratching it or breaking it. So how do we do that? Well, for that, in your lab drawers, you would actually have access to oil. So you'd have it in a nicely neat little beaker with a little vial that contains oil in them. And within that vial, you'd have a little glass rod. So I'm going to take it out of the actual beaker so I can show you the actual glass rod. Okay. And you can see the little bit of oil in there. Right? This is mineral oil. So how do we apply it is actually kind of a neat trick. So rather than going straight to the objective and crashing it, we actually go halfway. We end up having both objectives kind of split off to the sides. And then we can take the mineral oil. I'll show you how to do this kind of in reverse. Take a little dab of it and then apply it straight over the actual slide itself. Then we can close our little vial, place it inside our beaker for balance. And now, because there's oil on it, as you transition into the oil immersion lens, it shouldn't crash, it shouldn't break, it shouldn't do anything. So now we can actually see things at the highest magnification with these scopes, which is the oil immersion. So now we get up close. Now you can start seeing, as I adjust this guy, there you go. Now again, these cameras are not the best to use with the scopes, but they do give us a really good idea of what's under the actual slide itself, right? So there you have a bunch of little bacilli, some of them in like little diplo form or little strep form in there. And so this is part of what you're going to be drawing. So this is what's going to be provided to you in Canvas so you can actually draw those and place them into your lab manual and lab notebook. So that's part of the plan. So here's the oil immersion example of that. So now to do this in reverse, it actually takes a slightly different skill. Let me switch back. Since this slide now has oil and so does the lens, you don't actually want to go in reverse and contaminate the rest. So the way it's done is actually go in reverse and do that split piece, right? And now you can actually slide out the slide. That sounds kind of silly, I know. That way, whatever has oil in it is now taken off to the side. So I'm going to put this off here. And now you can actually go in reverse again, like we taught you before, from blue to yellow to red. And now it's safe to bring down your stage. And now you've officially observed your first organism under oil immersion over here. So now that we've kind of gone through the examples of part focality using the oil and more or less a quick review of using the scope, your job will be to take the images that we generate from this. They'll be available for you on Canvas, so you can place them inside your lab manual and your lab notebook. And again, like I said earlier, you should be able to use these so you can draw them in your lab notebook, use the actual grid that is part of it to measure things and actually do a little bit of algebra in there to kind of determine the sizes of what you're targeting in there, typically the sizes of cells. So that pretty much puts together all the labs that we wanted to accomplish today. So let's kind of make sure we have everything that we need to make sure we're ready for the next session. Is again, we need to keep one of our samples maintained, meaning we need to keep them alive. So that means either the broth or one of the previous plates, we want to make sure they last us until the next session. So again, as a reminder, you can always place them in the fridge as a way to slow down their growth. Okay. Now that being said, whatever you can save from the previous lab, that should be enough for the next one. If it's the TSB, remember you need to kind of resuspend it a little bit so you can pluck off some materials from there. Continue to use your fold scopes to practice your microscopy skills because we're going to now need those to use them in the next session. And don't forget that once you set up your lab 19, which is the selective differential media one, you're going to set it up, give it about 48 hours, and at least one of those plates should be growing. So you can take images and then write down the information that you have from there. That should pretty much put together all the labs for this particular session.